Greetings, Bio 321 students. So, what I want to do with this video is take you through the process of entering data and carrying out a two-way analysis of variance in JMP. And in the process of doing this, it'll be important that you have looked at the videos on how to keep your notebook. If you haven't done that yet, why don't you go ahead and do that now. Just pause the video and go do that. I'll wait here. Okay, now that you've done that, I assume you're back from watching those videos, and you've particularly watched Jen Chandler's video where she showed you how to set up a data set, right? So let's imagine we have an experiment where we are looking at the interactive effects, potential interactive effects of nitrogen and phosphorus. Okay, so we have presumably designed an experiment that is factorial in the sense that we have all possible levels of one factor with all possible levels of another. And for simplicity, I'm going to make this a 2x2 design here, where we have a low and high level of nitrogen, a low and high level of phosphorus, and we have the simplest possible design. Again, I'm just doing this to make this incredibly easy, not that it's going to simulate your experiment, which will have higher replication and perhaps more levels of your two factors. But remember that your two-way factorial has all possible combinations of those levels in your design, right? And so why are we designing this experiment? Well, let's imagine there's a river here, and there's proposed to put a livestock feed yard here, and we know that there's nitrogen runoff when you do that and you put it next to a river. And over here on this side of the river, this is really a river that's besieged with, we have a chicken farm besieged with problems, and the runoff from chicken farms is largely phosphorus. And so the local land zoning board is deciding whether to allow one or both or neither one of these two into the river, and therefore they're interested in the growth of some kind of organism downstream. So here's our organism that's getting potential runoff of phosphorus and nitrogen, and we've got low, which are presumably without the farm there, and high levels with the livestock farm, high levels of phosphorus with, anyway, this is why we're doing the experiment. So I'm concocting a situation here. It doesn't meet really the criteria of our biology science foundation solicitation for proposals. It's just for illustration purposes only. And now, if you've watched Jen's video, you know that we're going to have some column in your data set that might be, for example, plant ID. And of course, with this design, we're going to have 8 different plants that we're looking at. You know, maybe they're duckweed or something, right? And so we're going to have 2 different nitrogen levels. So we'll have a column of our data for one of our x variables, x1. This is our independent variable, right? And we're going to have 4 measurements that are made at low, right? And that's these 4 here in the upper part. And we're going to have 4 measurements that are made at the high levels of nitrogen. Likewise, for phosphorus, we're going to have 4 lows. Those are going to be down here, right? Because these 2 go with a high nitrogen level. And we're going to have 4 highs. So we have all possible combinations laid out this way, and we see that every individual is exposed to a level of nitrogen, a level of phosphorus, and we're going to measure some dependent variable on that. Now, in fact, with your experiments, we're going to measure multiple dependent variables. Well, you don't have to make separate data sheets. You can have Y1, Y2, Y3, as long as you're measuring these characteristics all on the same individuals, just make separate columns of those. For this, I'm only going to give you 1. So let's imagine, for example, I want to know, let's have 2, 4, 6, and 8, and I'm going to have 4 and 6, and I'm going to have 18 and 22. Okay, so this is some kind of sample y variable. It might be, number of lemna. We might have put in 1 lemna in each of the water samples with each of these levels, and we're going to count the number after so many days. All right, so this is our data set, and what we want to do is we want to put this into JMP, and I'm going to just quickly write down these numbers so that we get these numbers right. And okay, so how do we put these into SAS JMP? Well, the reason Jen told you to set it up this way is that this is exactly how it goes into SAS JMP. So you don't have to do any changes. What we're going to do is open SAS JMP, and we're going to create a new data table. All right, so let me expand it out here, right across my face. And we're going to, first of all, add columns. So the way you do that is just to click on the top, right? And now, oh, you want to see me, okay. Now we're going to give those columns names. And I'm going to call this one plant ID. I'm going to call this one nitrogen, phosphorus, and my Y variable. I'm giving them kind of shorthands here, so again, to keep this going along quickly. And I want to add rows to my data set. So I'm going to add 8 rows. Okay, everybody see how I did that? I found it under the rows menu up here, and I just said add rows. All right, and so I'm going to, first of all, add my IDs. 1, 2, 3, 4, 5, 6, 7, 8. All right, I have 8 plants. Next thing I want to do is add my my nitrogen levels. Now, I'm not going to put in numeric nitrogen levels. I'm going to put in a character. So I'm actually going to change that. The way I did that was I double-clicked on the top part, and this popped up. So my data type is character. And so now I'm going to put in my low, high. Right? Okay. And with phosphorus, it's going to be the same thing. I'm going to double click on it and change the variable to a character variable. And again, I'm going to do low, high, low, high. All right, now I put in my numbers, and what did I have? 2, 4, 6, 8, 4, 6, 18, 22. Okay, now my data set is in JMP, but I haven't told you what to do with it. Now before, when we did analysis of variance, we used a platform called Fit Y by X. And that was very nice. It was a simple platform. Fit Y by X is telling you there's one Y and one X. Now when it's more complicated than that, what we have to do is use a different platform called Fit Model. And it's pretty easy to use. You should double check that you actually have a little histogram next to your factors, your independent variables here. And when you set up your statistical model, what you're doing is you're saying, okay, I am proposing that y is a function of x, that my dependent variable is a function of nitrogen and phosphorus levels, and I'm going to construct the model based upon my analysis of variance hypotheses, right? So what did I hypothesize? I hypothesized there might be a nitrogen effect, there might be a phosphorus effect, but the effect of nitrogen might depend on the level of phosphorus. So I click on nitrogen and then shift click on phosphorus and cross the two, okay? So that gives n times p, it looks like. You know, when we talk about non-additivity, that's kind of what the interaction is about. Well, the times thing actually indicates that it's a multiplicative effect, not an additive effect. But n cross p is the way we say that, and so when we select the two variables, we hit the cross button, and that puts that down here. Now, I can remove any one of these if I make a mistake. Let's say I want to remove all that and start over. There's another way to construct this model. It's a little easier. You shift-click on these two, and then go to Macro and choose Full Factorial. And it does the same thing. Remember, this is a two-way factorial design. We've chosen two independent variables, so we can choose both of them and add them to the model. Now what we do is put our y variable up here, and we have to have a y or it won't run, and we just say run, and voila! JMP outputs quite a bit of gobbledygook here. And the important thing is that you understand where to get the information that's important here. So I'm going to shrink this down a little bit and see if we can pull up our little pad here and try to write an ANOVA table out of this. Okay, so where do we find our ANOVA results? Well, first of all, you can see some means here. Those are interesting to look at. And obviously, if you want all four means, they're over here. And, you know, I've given you an example where I gave the nitrogen level first and then the phosphorus level. So they are in that order. And if you do use L and H for your levels for both of your factors, you could run into this problem. You don't know which it is. But it follows the same order in which you put them in the model. Okay, so the first one is nitrogen and the second one is phosphorus if I want to ever plot these out. Okay, so how do we find our ANOVA? Remember how to make an ANOVA table? Let's go down here. Okay, we're going to make an ANOVA table. What goes in an ANOVA table? Our source of variation. That's the first element. What's the second one? Degrees of freedom. You're right. I know you remembered that. And then we have our sums of squares. By the way, if you need to do a little more background reading on this, look in the lab manual. It's there. We have our mean square. And we have our F value, which is, of course, the statistic of most importance. And then we have our P value, our probability of making a type 1 error, saying there's a difference when actually there's not. And we're looking for significance, quote unquote, would be where we have P less than 0.05 typically, unless we have set some other threshold level where we're going to recognize significance, which is not usually the case. Typically you'll just use 0.05. Okay, where do we find our results? We go down here to effect test. So click on that little triangle there, and now we can find our effects. All right, so now I can just find my sketchbook. But we have them. Okay, so here. So we have nitrogen effect. How many degrees of freedom? Well, we had two groups, so we have n-1 or a-1. Degrees of freedom equals 1. The sums of squares, 112.5. The mean square is the sums of squares divided by the degrees of freedom, right? Okay, so that is also 112.5 in this case only, because we have two groups, two nitrogen groups, two levels of nitrogen. All right, our F value is 32.1429. And our P value is 0.0048. So what do you think? Statistically significant effect of nitrogen? Hold on. It is less than 0.05, so you would think so, right? But we're going to talk about that for a sec. Phosphorus. Phosphorus effect, degrees of freedom 1. We have, what do I have? 180.5. Wow. And our mean squares, our sums of squares divided by degrees of freedom, 180.5 also. Okay. And our F ratio, 51.5714. And our P value of 0.0020. So what do you think? Significant effect of phosphorus? It's less than 0.05. Yep, you would think so. Well, hold on. We need to look at the nitrogen by phosphorus interaction, which we typically write that way. I could write out the words, okay, but I'm going to give it abbreviation for now. We have 1 degree of freedom there, and we have 80.5. It's our sums of squares. Mean square is sums of squares divided by degrees of freedom, so it's 80.5 also. The F value is 17.2857. And the p-value is 0.0142. Wow, that's less than 0.05 also. Okay, now we're not quite done. We have one more row to put in a complete ANOVA table, and that's the error term. And we find that actually by going back up to the analysis of variance table up here, where we see that we had, we see that we had 4 degrees of freedom for the error. We have a sum of squares of 367.5. And we have a mean square. I'm going to have to get my calculator for this one. 367.5 divided by 4, 91.875. Oh, sorry. Sums of squares is 14, isn't it? I need my eraser. I'm going to have to pull this up. Sorry about that. I just, I don't have my glasses on here. There we go. My eraser. All right, so we have sums of squares of 14. I knew something was funny there. Oops, I better get my pen back. (14.0) divided by 4 is 3.5. Notice it gives you the mean square up there. I thought it did. That's why I knew it was wrong. And so that's as far as we go. There's no F value associated with the error term. I know it looks like there is here, but this, that's weird. F ratio, probably. Anyway, this is a whole model. I know it's just showing a part of the thing there. But anyway, there's no F value associated with the error term because there's no error term for the mean square, right? Remember what the F ratio is? It's the ratio of 2 mean squares or 2 variances. This, by the way, the mean square is the variance that's distributed to each of those things. And this 17 comes from 80.5 divided by 3.5. And the 51 comes from 180.5 divided by 3.5. And the 32 comes from 112.5 divided by 3.5. So I'm drawing these arrows to show you what the denominator was for F in each case. The numerator is here, this mean square, and the denominator for the ratio, remember F is a ratio, so the numerator is here and the denominator is here for all of them. That's the error. Okay, so how do we interpret this? Since this is an important component here, and really an important thing to think about. So what we have is a case in this instance where, well, I guess I should look at those means. So I can look at it down here. So our low mean, so if I were to plot these out, and I have nitrogen and phosphorus, low and low, that mean was 3. I'm just going to plot this out. And then when we went to a high phosphorus level, low nitrogen but high phosphorus, what happened? So we add phosphorus and it goes up to 7. I'm going to give adding phosphorus being a dashed line like that. Okay, and then we go back to if we go to high nitrogen but low phosphorus, what we end up with is a mean of 5. Okay, so we're just looking at the phosphorus effect with the different colors here. And if I go to high nitrogen and high phosphorus effect, then what we end up with is, I think I have 19, a big number. I built in some non-additivity here. So what you can see is, this is the effect, that difference between the means is the effect of phosphorus at low nitrogen, but this difference here is the effect of high phosphorus at high nitrogen. So you can see the effect of phosphorus on lemna growth depended on the nitrogen level. Okay, so that's one way we could write this interaction, and we would support that with F equals 17.2857, P equals 0.0142. All right, so this is a results-like statement that we would make. We would then go on to say, in addition, or to clarify, we would say that with phosphorus, with high phosphorus, let's make it clear, high phosphorus having a greater effect at high nitrogen than at low nitrogen. In other words, and then we would reference our figure, figure 1. Here's our figure 1. And the figure 1 legend would say something like dependency of lemna growth on nitrogen and phosphorus. All right, so the figure legend just describes what's in the figure. We put our y variable, we would have our units here, make sure we do that. And we would also make sure, of course, to put standard errors on our histogram so we could compare those means, right? I'm trying to give you a bit of good practice here. Of course, we would all make this very neat as well. But we would have these kinds of statements about our ANOVA table. And our table legend might say two-way analysis of variance for lemna growth data. Something like that. Okay, and then we have a complete table there. We would reference the table. And by the way, typically you would either reference it this way or reference it with the table and not both. But I'm just showing you how you could do it either way. Now, I said, okay, now what do we go on and say about phosphorus and nitrogen? This one is actually kind of tricky in this instance because clearly our interaction, which we look at first, says that there is a dependency of the effect of phosphorus on the level of nitrogen, right? So how can we then go on and say there's a phosphorus effect? Well, what we have to do is look at our diagram and see, well, was the phosphorus effect always consistent? And in this case, I would say yes. So the phosphorus effect was that it stimulated growth of lemna, right? It was always that way. We can look at the overlap of these error bars perhaps and make sure those didn't overlap, and we would say, okay, there was a stimulation at low nitrogen and there was a stimulation at high nitrogen. So regardless of the nitrogen level, there was a phosphorus effect. It's just that we also say first that the phosphorus effect was greater at high nitrogen than at low nitrogen. All right, so if you have to stop and listen to this a few times, don't worry, that's fine. That's okay. Now, what about the nitrogen effect? Let's talk about the nitrogen effect here. So, was there always a nitrogen effect? Well, let's look what happened to nitrogen. At low phosphorus, really there wasn't a nitrogen effect, was there? It didn't go up significantly. But it did go up significantly when we look at these two means, So we're comparing low and high nitrogen at high phosphorus level. Yes, they went up. but they didn't really go up significantly when we only have low phosphorus. So would I talk about that nitrogen main effect here? No, I probably wouldn't, because it's not consistent. It's not always the same. It depends on the level of phosphorus whether we had a nitrogen effect or not. Now that's a subtlety, okay, and we're not going to penalize you too much if you talk about main effects even though the interaction was significant, because there is some judgment involved. I'm judging here that the effect of phosphorus was consistent, and I'm doing that by seeing that it was always positive and that the error bars don't overlap. But the effect of nitrogen was not consistent because these error bars between this particular phosphorus level did not. They did overlap, and so there wasn't a difference there. So it wasn't a consistent nitrogen effect across all phosphorus levels. There was a dependency. All right, so, you know, can the statistics lead you astray? A little bit, and you have to use some judgment once you have found a significant interactive effect. You do that first and interpret that, and then you interpret the main effects above that only after that, and you may or may not want to talk about them in your results statement, depending upon whether the effect was contingent upon the level of the other factor, just like I explained. So here with phosphorus I would talk about it, but with nitrogen I wouldn't. All right, I know that's a little bit of complexity there, but hey, you're ready for it, right? You understand two-way ANOVA now. Okay, so two-way ANOVA, just to review, we put it in SAS JMP, and we're going to use the form kind of like Jen did in her example, and we're going to get an output like this, and we're going to take the effect tests and construct an ANOVA table. We're going to take our means here. I don't know if I copied those quite correctly, but anyway. We're going to take our means and we're going to plot them in a nice figure with units on both axes, error bars, and so forth. And then we're going to talk about them in results as I've shown you how to do. Okie doke. By the way, obviously I'm prepping you for a quiz about this, which you might see in class next week. So let me explain a little bit too. I mean, the reason we're kind of hammering these statistics home is that this is the essence of what you're going to do with your data in the end. You really need to know how to do this with your own data. We're going to ask all of you to do this, and you're going to have data exactly in this form if you're doing a two-way factorial design. So we want you to know how to do it and how to go through all the steps. You may have to watch this video a couple times before you'll get it down. You should practice it with a data set that's like your data set, okay? But this is sort of the essence of how you're going to know what your results mean in the end and how you're going to interpret it scientifically, because it all goes back to answering the question, does the effect of one factor depend on the level of the other factor, right? Which was the whole basis for the entire experimental two-way factorial design that you designed for your particular experiment, and you're going to answer that question, and it will be significant or not depending upon what your results are going to say. Now, the other thing I want to talk about is sort of biological significance versus statistical significance. You want to look at the size of those effects that you're seeing in the graph that you produce. So for example, you might even mention that the effect of nitrogen was to increase lemna growth 20% at low phosphorus, but 100% at high phosphorus. So try to give the reader an indication of the size of your effect. That's the biological effect. Sorry about my bird clock. Anyway, I suggest you talk about it in those kinds of terms so you can help your reader interpret the biological significance. And don't dwell on the statistical part of it. It does matter in terms of whether you assert that there is an effect or not, or whether there's an effect that depends on the level of another factor. It's going to, your statistics are going to back that up, but use them sort of parenthetically. Refer to a table or put parenthetically F equals, P equals, and talk about the biology. Because the biology is what's important, right? It's not the statistics that are important. The statistics will come out however they come out. And then you make appropriate biological statements as a result of that. All right, I'll stop preaching now. Talk to you later.